I'm going to talk today about a part of Wisconsin history that's not nearly as widely known as some of the other things we've talked about this semester. If you ask the average man in the street or woman in the street what happened at Wisconsin in science, they'd certainly name vitamin D as one of the things. They would probably name Carl Paul Link and the discovery of warfarin another. But today I'm going to talk about a development that took place here during a very exciting period of American history, that is the period of the Second World War and the needs of that war, and the role that Madison's microbiologists had essentially in developing penicillin from a curiosity, which it was in 1940, to a useful and available drug by 1944. Wisconsin has always had, the agriculture school has always had an interest in in fermentations, which are reactions in which you take some inexpensive, usually, source of a sugar, put it in a closed vessel where there's no oxygen allowed, and let the bacteria that are on it work on it so that they convert it into an acid, which has the effect of preserving the vegetable matter and making it last for weeks or months or years. Pickles are fermented cucumbers, sauerkraut, soy, yogurt, those are all fermented things. Beer and wine, of course, are fermented. Industrial laboratories can take cheap things like corn steep, the juice from corn, and convert it into expensive things or relatively expensive things like ethanol and butanol. The cheese industry involves basically a fermentation process, same for sausage. But today I want to talk about penicillin, which turns out can only be made successfully by a fermentation, and it depended on the availability of techniques that had been developed at Wisconsin for these very practical reasons. We've talked in class now about two kinds of test organisms to see if you can detect something that is needed for a diet. One of those test organisms was the whole cow, where we saw the discovery of the vitamins took several years because the life cycle of a cow is that long. And cows, of course, turn out to be expensive and hard to house. And as a result, the nutritionists here changed over to using rats as their microbial, as their test organism. And they're in every way easier to work with in the laboratory. You can get much better statistics with them, so they were clearly important. But there's one more step down in size we can take, and that is the use of microbes, bacteria or fungi, as a test organism that you can feed to see if you can find components of the diet that they require. Esmond Snell and Frank Strong were members of the faculty here in the 1940s and 50s, and they recognized that if you had a bacterium that required some component of its diet, that is, it couldn't make something like a vitamin, then that bacterium's growth would depend on your adding back the right amount of the vitamin. This actually shows two kinds of experiments here. In both cases, what's being measured is the growth of bacteria. This particular one on the left is lactobacillus. The one on the right is leishmania. But in each case, what they're showing is that if you start with a bacterium and add various things to its growth medium, the growth of the bacterium will depend on what you add, just like the growth of the dairy cow depends on what you added back. So if you had a bacterium that was in a rough medium that provided a carbon source like glucose, provided a few inorganic things like magnesium and phosphate, and then you asked it to grow, it wouldn't grow unless you added something back. And the something could be an ether extract of some yeast material, or it could be any of a number of things, but the stuff you added back had an effect of making the cells, the bacterial cells, grow more, and that extra growth was a measure of the value of the stuff you were adding as a dietary supplement. Here's a cleaner experiment in which one of the investigators, it was actually Snell, has got a bacterium that he knows is completely dependent on the addition of vitamin B12. Here, cyanocobalamin is another way of saying vitamin B12. That bacterium only grows if it's provided with that, and it grows as much as it's provided with the vitamin. So if you look along the bottom scale here, this is the amount of vitamin B12 added. Along this scale, it's just essentially a measure of the number of bacteria that grow after an overnight incubation. And what you see is that there's a nice linear dependence of growth on amount of vitamin B12 added up to a certain point where it begins to bend off. That means that you could use the growth of the bacterium as a method of measuring how much B12 there was in any sample you wanted to add. And it's worth noting here that this is an incredibly sensitive method. The amount of vitamin B12 being added here is picograms, an amount you can't begin to see in the palm of your hand, but it gave an easily distinguishable, easily measurable result here that can be so easily quantitated. What happens is you take a set of 10 or 100 or 1,000 test tubes, put the basic minimum that's not adequate in there, inoculate them with the bacterium, and then add to each of the tubes something that you're trying to test. And this standard curve allows you to figure out from the amount of bacterial growth how much vitamin B12 there was in each sample. Tremendous advantage over either using cattle or using rats, and allowed for much faster testing of foods, for example, to see which foods were rich in one thing or another. This was used very widely after Snell and Strong developed it, and it was used here by Snell to isolate another of the vitamins, pyridoxal phosphate. So microbial tests, microbial fermentations, that's the first player in this story. The second players is a whole team that were here over a long period of years, but mainly from about 1925 until about 1970, this whole gang here. They were from a number of different departments, including bacteriology, biochemistry, botany, and pharmacy school. These were people who had a variety of interests, but they all had in some way a dependence on the use of fermentation in their studies. E.B. Fred, that we've talked about before, was the chairman of bacteriology, the dean of the graduate school, and the president of the university. Elizabeth McCoy was the bacteriology prof, who I think was the first tenured science woman in Madison. Fraser and Sarles were also, and Raper were in bacteriology. There was a group in biochemistry, and maybe surprising to you, but not so unusual, there were even botanists who studied fermentations. So when the need came during at the beginning of the Second World War to develop penicillium strains that produced lots of penicillin, the departments were ready. They had people who had spent half their lives studying bacterial growth in large cultures and were therefore ready to step in. So a third player in this story here is the people who chased the silver bullet. There was a period between about 1900 and 1920 when the hope was that you could find some chemical that you could add to a bacterium or to a person and kill the bacterium. And the reason for this hope was that it was clear from microscopy that there were various things, dyes, you could add to various bacteria, and certain dyes only stuck to certain bacteria. And the hope was that you could find a dye that stuck to the bacterium and, better yet, killed it. So a whole large range of compounds that had become available as oil was being fractionated in Europe and studied in lots of organic chemistry labs. These compounds were tested to exhaustion practically. There were also people who essentially took a guess at what might be toxic to bacteria, and Paul Ehrlich was one of them. He was a world-famous scientist, one of the top physiologists of his day, won the Nobel Prize for earlier work that I'm going to talk about. But he decided that there must be some way to target bacteria with compounds that were known to be poisonous in some way to see if they would be more poisonous for bacteria than for the humans that had the bacterial disease. He started with a group of compounds you probably can't see, but they're arsenicals. This one has 3 arsenics in it. This one has 5. They were intended to, and they turned out to be effective against syphilis, which was a very common disease at this time, and serious disease in the sense that it could kill you. And Ehrlich and his technician started out making compounds that were related to these arsenicals, one compound after another after another. They had tested 606 of them before they came to one that had the effect of killing bacteria in the laboratory and not killing the people when you fed it to them. It was a widely prescribed drug during the period that it was all that was available, and made Ehrlich especially popular among the people who had syphilis, I guess. At the same time, Ehrlich was a German. At the same time in France, the French scientists were doing the same thing. They were trying to find compounds, some derived from dyes, that would stick to a bacterium and kill it. And they found that a lot of the compounds that were in principle suitable for this purpose were not usable because they were not soluble in water, or they didn't stay in the blood system long enough to be useful. So they started making derivatives of these things, and the derivatives were sulfonamides. You needn't worry about the structure of these things, but sulfonamides are simple derivatives of these things that are more water soluble. And they discovered the French groups, they're not represented in this slide, discovered that they did get some compounds that had promise. It turned out that it wasn't the compounds that were promising, it was the sulfonamide that they added to make them soluble. And it took a long time for them to figure that out, but when they did, they began to market the stuff. And so there was a German product and there was a French product. The German product came to be called Penicillin. It was a sulfonylamide. Gerhard Domach was the researcher who did this work, and he eventually won the Nobel Prize for it. But in 1939, when the prize was offered to him, he couldn't accept it because Hitler was in power. Hitler had been annoyed a couple of years earlier when a pacifist in his country had been given the Nobel Peace Prize, and he told his people that from then on there were no more Nobel Prizes for anybody. When the war was over, Domach got this. But Penicillin was his compound, and it was the only thing really available for other diseases than syphilis. There's a great book that I recommend called The Demon Under the Microscope, which is about Domach's search for these compounds, about the fight between the French and the Germans, about the effects of the rise of the Nazis, and it's a nice combination of history and science. You all know the story of Alexander Fleming, who was working in England at this time, in the United Kingdom, and who made an accidental discovery and then was smart enough to follow it up. Fleming was a bacteriologist, basically. He worked in the laboratory, hospital laboratory in London. And he left one weekend without cleaning up his mess, left behind a few petri dishes that he had streaked with bacteria. He came back from vacation and found a plate that looked like this. In this case, what's happened is that this mark represents the bacteria that were being streaked onto the plate, put onto something that gave them all the nutrients they needed, and they grew and they made thick mats of bacteria, billions of bacteria here. But a mold, apparently from the air, had fallen onto his petri dish, and given the long vacation that he took, the mold had the time to grow. And what he noticed was that around the mold there was this region where the bacteria didn't grow. And he correctly reasoned that the mold was putting out something that was diffusible and soluble, and diffused out far enough so that in this region it was a sort of a no bacterium's land. That was a complete accidental discovery, but he quickly recognized the potential of whatever this stuff was, and he began a period of about 9 years of research that he carried out more or less by himself and more or less unsuccessfully, trying to figure out if there was a way to get this to happen on a larger scale. The problem with this kind of an experiment is that the amount of stuff that was being produced here is minuscule, and you have to somehow figure out a way to make more of the mold so it'll make more of the stuff so you can characterize it. He essentially gave up after a period in which he tried all kinds of tricks to get the mold to grow. Molds typically grow on the surface of something, and the way you get a lot of mold is not to have a big jar, it's to have a lot of flat space with a lot of surface area. And he tried all kinds of things to provide that. He failed, and by 1939 or so, had essentially stepped out of the business. But this was the time the war was beginning, and everybody realized on both sides that a drug that would kill bacteria, stop infections, would be critically important. Here's a thing from the newspaper that shows that in World War I, of every 100 soldiers receiving medical care, 8 died, and most of them died, in fact, from infections, not from the wounds themselves. Whereas in World War II, it turns out that only 4 died, and so there's a very significant military advantage to having penicillin. Here's an ad from a newspaper. So the governments of the UK and the United States got together. The UK was being bombed by this time, and the laboratories were not safe. They couldn't be sure that they could scale up a big operation without having it all blown apart. So these several investigators from Europe to the United States. They met with a group of executives from the large pharmaceutical houses in the States, and they decided to carry on what was essentially a Manhattan-style project in developing penicillin. No limitation on money, no limitation on people. The project could pull people from wherever they were to get them together, just as they did in Los Alamos to make the bomb. And from the period of 1940, when there was almost nothing in a soldier's kit except sulfonamide, until the end of the war, there was a huge increase in the ability to make this stuff. This is a monument to Fleming's unsuccessful efforts to make lots of penicillin. After the war, before his death, he won all kinds of medals for his contribution. And in this display of medals, there's an interesting artifact here, which is a hospital bedpan. When Fleming realized that the penicillin grew on the flat surface, he figured out that the only large number of flat surface vessels around a hospital were all the urinals, and so he gathered a whole bunch of them and inoculated a whole bunch of them. Still couldn't get enough penicillin to be anything more than a curiosity. These are the people who took it on to the next level. Ernst Chain, and Howard Florey, both working in the United Kingdom, also at the University of Oxford. This was a chemist. Chain was a chemist. Florey was more of a medical type, but the two of them both recognized the need to cooperate and to find some place where large-scale fermentations could be carried out to make lots of penicillin. They came to the country. They talked to various companies here. As far as I can tell, none of the companies excluded themselves. They all jumped in with this, even though there was no promise of profit. But what they did was to divide up the work, and the work had to be on several issues that would each impinge on the final amount of penicillin they could get. One was, what kind of stuff should we grow the mold in? The second is what its physical state. Should it be a solid, a gel, a liquid? What temperature was best? Molds are notoriously different in their behavior in different conditions of temperature. There was an important issue about finding the best strain of Penicillium, the mold that this is produced by. And then there was the always present problem that even if you got a strain of Penicillium that would really pour out penicillin, you had to be able to separate the penicillin that they made from all the other stuff in the bacteria, some of which might be toxic, and get it out of there so that you could get pure or relatively pure penicillin. This is the mold. It's a sporulating mold like all of them. And there's a whole family of these things, different in small details, any one of which might have been the good one for making penicillin. There's another book that you might enjoy called The Mold in Dr. Florey's Coat, and it's an account of the fact that as the war wound up, there was real worry that the existing progress on penicillin production would be lost by a stray bomb, and so, or an arrest. So Florey had a lab coat that he soaked in penicillium, the mold, and he said that any time they came for him, he's going to put his coat on and take off. So, the Wisconsin connection, it begins here with a guy who was a professor in the Department of Bacteriology, Kenneth Raper, who was a mold expert and therefore a perfect guy to get involved in a project like this. For this Manhattan-style project, the government picked just a few laboratories, one of which was in Peoria, Illinois, which was called the Northwestern Regional Research Lab, NRRL. And they pulled into that laboratory Ken Raper and a number of other people who had the right set of skills to do what they were going to do. Raper came back here after the war and had a very distinguished career studying slime molds, and made the slime mold dictyostelium the preferred target for developmental studies for many years. In Peoria, they had several ways of coming at the problem of getting better production of penicillin. During this period, they wanted to sample as many soil samples from as many places as they could. So the Air Force, actually the Army Air Force then, was told that anytime a pilot touched down in a foreign country, he should scoop up some dirt and bring it back. They actually did this. They collected samples from all over the place. But in Peoria, there was a farmer's market, and Ken Raper, this fellow I'm talking about, had an assistant, her name was Mary, whom he sent to the farmer's market every day and told her to buy the rottenest cantaloupe, the rottenest vegetable she could lay her hands on. She became known as Moldy Mary. And this painting of Moldy Mary hung in the bacteriology department for many years. I think it's still there someplace, probably not in a place of honor anymore. But in any case, she got the winner. She found a cantaloupe that was really rotten, and it produced penicillin at a rate much higher than the other molds that had been isolated. So that became the starting material. These people all had experience in strain improvement, that is, finding a strain from nature and then cultivating it in the laboratory to find the best conditions to cultivate it, the best strain. In some cases, they used sort of raw, they went at this with blind force, not careful attention, but just trying to find from among the thousands of cultures that they had one that was going to be good. And the result over time was that these people did get better and better and better strains of penicillium, which means they made more and more and more penicillin. Some of these people we've talked about, this one was a key member of this group of fermentation biologists. When we talked about Josh Lederberg, I told you that his instructor was Tatum. Tatum had grown up in Madison and was actually the graduate student of W.H. Peterson, so there was that long connection. Marv Johnson had also been a student of Peterson. Burris had worked with E.B. Fred. Perlman had worked in industry as a in a pharmaceutical industry, but he was back here and participated in the large-scale fermentations. And the net effect was a production of better and better strains of penicillium. These are the strain designations, and the ones that are designated WISC are the ones that came from Wisconsin. And of course, what you see is that they all, eventually, the winner, this strain down here, which makes the most penicillin, is derived from Wisconsin stocks. Here's an illustration of how successful they were in increasing the production and lowering the price of penicillin. Here we have plotted on the left an exponential scale, the amount of penicillin made over the years. And what you see is that in 1935 there was none. 1939 there was 10 to the first, and the units are, what are the units? tons, I guess. Anyway, the stuff cost $11,000 a kilogram at that time. Another order of magnitude in another year, another order of magnitude, and another, and another. So they actually took the yield of penicillin up by 5 orders of magnitude, made it possible to get from 1 milliliter of the growth medium 50,000 units instead of the 4 units that they got in the original preparation. So they had essentially taken the stuff from being a laboratory curiosity, only sufficient in amount to treat 1 patient, to an amount that by the end of the war, by Normandy, actually could treat everybody. Huge success.
And they grew this stuff on what to produce it?
The basic medium, I think, was corn steep, which is dead cheap stuff, and it was always available in huge amounts. But it meant changing the inorganic ion concentrations. It meant changing the pH, and they finally found the right concentration. So that's the end of the war story, but the story didn't end there, because there were still questions, important questions about, for example, how penicillin worked. And that question was essentially answered here by 2 people in 2 different labs with 2 different approaches, Marv Johnson, M.J. Johnson in biochemistry, and Jack Strominger in the UW pharmacology department. Johnson and one of his graduate students, whose name, of course, I can't forget, or can't remember, Johnson, can't remember. The 2 of them took bacteria that were susceptible to penicillin, treated them with penicillin, and looked to see what they spilled out when they spilled their guts. That's what happens to E. coli or other bacteria. They break open. Strominger went at this looking for the enzymes that were inhibited by penicillin, and they both came to the conclusion that there was a pathway to cell walls in bacteria that was interfered with by penicillin, and that the cell wall target was only a target if the bacteria were actually growing. If they weren't, or they weren't trying to make cell wall, then the penicillin didn't hurt them. So it kills living, growing cells. This is a picture through a microscope that shows what I've just said. When the cell wall is trying to expand to make the cell longer, the cell cuts the cell wall up into little pieces so there's room to put in new pieces. And at that point, if you treat with penicillin, they never can put in the new pieces and they spill their insides. Here's the insides. We talked about Josh Lederberg, and you remember that he introduced several techniques for studying bacterial genetics. In fact, he sort of invented the whole field of bacterial genetics. He studied E. coli, and of course, E. coli turns out to be one of the many bacteria that is sensitive to penicillin. And he used penicillin as a laboratory tool. When he understood that penicillin only killed growing cells, he knew that he had a technique that could separate from a very large number of normal cells that were growing from a very small number of abnormal cells that were not growing. And he was able thereby to rig things so that he could get bacteria that had everything they needed in life except the inability to make 1 amino acid. And that means that if you grew them in a medium that didn't have that amino acid, they wouldn't grow. All the other cells would, but not these. And if you treated with penicillin, all the other cells got killed, but not these. And now you had a mutant that was defective in the synthesis of something. So he made hay with this discovery, and it was really a very critical part of the development of bacterial genetics. There was also an early recognition of the fact that it would be too good to believe that this toxin for bacteria would always be useful, and it turned out fairly early on that strains of bacteria developed that were resistant to the antibiotic for various reasons. And we now know that there are strains of bacteria that are resistant to 4 different antibiotics, and they are superbugs that are extremely dangerous and cause lots of concern in the hospital community. Tugger was a member of the pharmacy department here. He had worked at the Lederle laboratories and had long chops as a lab supervisor. And he studied the antibiotic aureomycin another mold-produced antibiotic. David Perlman, who was for a while the dean of the graduate school, the dean of the pharmacy school, also came from industry, Hoffman LaRoche, Merck, Squibb, in 1967, and his contribution was the studies of neomycin and tetracycline, two more drugs that were discovered from nature. Then there were these two. Julian Davis was a colleague of mine in biochemistry who is now 80 years old and still working every day in the laboratory at the University of, anybody know where he is? British Columbia, University of British Columbia. And the other picture on the right is Bernie Weisblum, who has lived in the laboratory for a long time, and the two of them both targeted streptomycin, another antibiotic, asking how it works, and they both showed that it works on ribosomes, and we know in quite grisly detail now how it works on ribosomes. So there's been a lot of contribution to the use of medical antibiotics by Wisconsin scientists. This is a moral tale that I want to tell you, that's unfortunately too common. Selman Waksman won the Nobel Prize in 1952 for the discovery of streptomycin. This is him on the right, of course, the old guy. This young guy was his postdoc, who worked in the basement of Waksman's laboratory, screening soil sample after soil sample after soil sample, and he actually found streptomycin. It was he, not the boss, who found it. His name was Albert Schatz. So I said, this guy won the Nobel Prize. This guy disappeared into obscurity for many years, I think almost 50 years, until a woman, whose picture is on the front page of this book, was saved by streptomycin. She had a disease that was going to kill her, and streptomycin saved her. And she was so grateful for this that she actually started to look into the history of the discovery of streptomycin, and she recognized that the graduate student had really done it, the postdoc, and she wrote a book, Finding Dr. Schatz, described him at the time when he was young, and at the time when he was already retired from his career. And it should have made, what's his name here, turn over in his grave to realize that he'd been exposed. Some of you may remember a movie. I don't see many heads nodding, but this is the Third Man's Theme. And it was a movie that came out either right at the end of the war or a few months or years afterwards, very soon after the war. No, I want to stop this. Which had three very well-known actors, Cotton, Orson Welles, Trevor Howard. It was a story about a crooked doctor who, during the Second World War, when penicillin, before it became readily available, was given very small quantities of this precious stuff to treat children in a children's orphanage. And instead of doing that, he diluted the stuff for the children and sold the good stuff on the black market. And the story was about him, and the only reason I show you this is to make it clear that there was a time when penicillin was life-saving and mighty precious. Now, a little bit more about penicillin. The penicillin that was used in the Second World War, the stuff that was made by the ton, was of an unknown structure. But this woman, Dorothy Crowfoot Hodgkin, working in the United Kingdom, was a crystallographer who used X-ray diffraction to determine the structure of complex molecules. And one of the first molecules she tackled was penicillin. She discovered that it had this structure, and the structure made it very clear why some bacteria were able to escape the drug and others weren't. And it also was the beginning of a very distinguished career in which she solved many other structures, more complicated structures, And for what she, pardon?
She was an ultrasound.
Yes, yeah. Now, one last related story here has to do with how cells become resistant. And I hope I'm telling you something you already know, but just as a reminder, if we have a bacterial cell here in yellow that is sensitive to a certain antibiotic, there are 3 ways that the cell can escape that antibiotic. Can't find my cursor. So the one way to escape is to have a little pump in the cell membrane so that if the antibiotic gets in, you just pump it back out. The second way is if you have a bacterium, an antibiotic that gets in, you have an enzyme that breaks down the antibiotic, and now those pieces that left over are no longer noxious. And the third way is a little bit like this, that is, there's also an enzyme, but the enzyme, instead of breaking down the antibiotic, alters it to make it harmless. So the enzyme makes a small chemical change in it. Any of those 3 things can make it impossible to kill the bacterium with penicillin or with an awful lot of other drugs, antibiotics in general. And so the pharmaceutical industry is stuck with the problem of finding ways around this. It's a much worse problem than one might imagine at first glance, because it turns out that once one bacterium has discovered how to be resistant to a drug, it can pass the gene or genes that allow it to be resistant around. They pass from one bacterium to another by any of 3 processes. One, conjugation, when there's actually a transfer of DNA containing those genes. One, transformation, when the bacterium donates DNA to the medium, which then penetrates the cell. One is transduction, in which a phage, a bacterial virus, carries in the piece of DNA that makes the cell resistant. And it turns out that this is a very common process, and it's the reason so many bacterial strains now, especially those that cause serious clinical problems, are resistant not merely to one antibiotic, but to the whole raft of them. I don't want to offend anybody, but I think this is sort of a funny cartoon. So let me read it to you. TB, my God, are you sure? Afraid so, but we caught it early. So my prognosis is good? Depends. Are you a creationist? Why, yes, I am. Why do you ask? Because I need to know whether you want me to treat the TB bug as it was before antibiotics or as the multiple drug resistant strain it has since evolved into. Evolved? Your choice. If you go with the Noah's Ark version, I'll just give you streptomycin. What are the newer drugs like? They're intelligently designed. So we have now a sort of continuing war against the development of more and more resistant strains, and this actually is a very serious concern to people. There are some pockets of geographic areas where superbugs are very common, and they're causing real concern. The pharmaceutical industry has always kept in reserve one drug that hasn't been used clinically, so that no bug has had a chance to get resistant to it. But the availability of those things is not going to be there forever, so it's going to be a problem for the future. Lots of possibilities for natural products chemists, lots of possibilities for the kind of people who look for bacteria in the soil, and lots of challenge in the future. I think this is the, yes, that's the end of my slides, and I'll be glad to take questions if you have any. Yes?
I heard of the extensive usage of antibiotics in feeding livestock, not just for curing purpose, but for supporting their growth. If this is the case, how do the antibiotics support their growth?
Yes, if you didn't hear the question, he's pointing out that livestock, beef cattle in particular, are fed large quantities of antibiotics in the diet. Not only or not merely to block bacterial infections, but because for some reason antibiotics in the diet make beef cattle develop more muscle and they're heavier. And the trouble is that the antibiotics that are fed in the diet don't disappear. They are still in the meat that you eat. And both the humans who eat these things and the cattle who got them in the first place provide a place for bacteria to get acquainted with the antibiotic and become resistant to it. And this is it's a serious problem. There's some, I don't know the legal situation, but there's been lots of motion towards making this illegal in some places, feeding feedlots with antibiotics. Okay.
I'll ask a question.
Yes, Tom.
The dark side of this, there was a professor, Ira Baldwin and Joe Swanson's father, who you may have talked to, who were involved in bacteriological research during the Second World War. Does that relate to what you've been talking about here tonight?
Yeah, Thomas pointed out that there was a Wisconsin contingent in Washington during the Second World War charged with developing either anti-bacterial warfare or bacterial warfare agents. Ira Baldwin, who was the dean of the dean of the College of Agriculture, was the head of this program. It was in Fort Detrick, Maryland, yeah. And it was a big program. It was not advertised around here, but I've read his biography, and he spent several years running this program. The way it's presented in his biography is that they were afraid that they would encounter biological warfare, and they needed to be able to counter it. So it wasn't so much about antibiotics. It was about things like anthrax that could be weaponized and used to wipe out populations. Yeah, so I've noticed that nobody says much about this in his official biographies, but I mean, it was a fact. He was there. Yes?
And I have a question. When you showed us the cartoon, it looked like a Bloomsbury cartoon.
Yes.
About the penicillin. You said that this is a real issue in some parts. You inferred in some parts of the world. So what precisely are you talking about amongst populations? You are not talking about, for example, the thing that comes to my mind, unrelated to this, is that contingent of American parents who don't want their children to have vaccines.
Yeah. Yeah, that's a different issue.
That's a different issue.
Yeah.
So what are you talking about when you say that it's real?
There are foci, geographical places where, for whatever reason, the superbugs that are resistant to 4 different antibiotics become very common in the hospital. I'm sorry that I can't tell you where this. It's a medical problem. Yeah.
People who say I don't want to have higher.
No, that's not the problem. But the problem is that if you go into a hospital, there's a good chance you'll get infected with a bug that they can't cure, and you'll die.
MRSA, is that the name of the drug?
Pardon?
MRSA, is that the acronym for the?
Yeah.
Yes, that's right. Yeah. Mm-hmm. Okay, it's almost the weekend. Hooray.